Thank you very much for the invitation to explain our work. It has been done at the National Center of Biotechnology by the Biocomputing Unit that is led by myself and Jose Maria Carazo. So what I will explain today is the use of Scipion as an ecosystem for cryo-electron microscopy. So I will just focus in cryo-electron microscopy, but it is a more general system and you can address other problems in structural biology. So Scipion itself is a workflow engine for structural biology. So it does orchestrate the execution of workflows that are somehow represented here. So when you have to solve a problem computationally, you typically divide that problem into different steps, and the output of one step is the input for the next one, and then Scipion is the orchestrator that allows this execution of the different steps. So it doesn't compute anything by itself. So it only calls external programs to do the image processing in this case. And what it provides is a layer of reproducibility. So in principle, up to the random initialization of the algorithms. But up to that, if you re-execute the same process and the process is deterministic, in principle, you should have exactly the same result. It provides you traceability because you know exactly which steps were performed with which parameters. You have all kind of analysis to see what was the output of that step. It integrates over 130 packages, and a package for us is a suite of programs that can do a specific task in this domain, and more than 1,000 protocols. So a protocol is, each one of these boxes is called a protocol, and you can combine them in any way that you like, so that you can construct very flexible workflows adapted to just your specific problem. It integrates well with clusters, high-performance computing, cloud computing. It allows the definition of the workflow manually, so you can add the steps one by one, or if you have a predefined workflow, you can execute this workflow fully automatically. And very important for us is what we call consensus algorithms. So some of these boxes compare the output of other algorithms that are supposed to compute the same thing, and that this consensus allows you to identify which parts are in common, which are the common output, and which outputs are not so common. So this is the outline of the talk. So we will start talking about what is the Scipion size in terms of usage and developers, and then specifically the topics for cryo-electron microscopy. So all our code is in GitHub. And if you look for Scipion in GitHub, we have 300 repositories. So some of them are software, some of them are also documentation. And overall, we have over 100 different packages, so different software packages. As you can see here, there are 95 developers registered in Scipion, so it is not only our lab, but many other developers around the world contribute to Scipion. The number of projects per country, because we are in Spain, many of the projects are coming from our surrounding, but you can see that the usual countries for research, France, United States, United Kingdom, they also use it quite extensively. So a project is one project corresponds to one structure. So it is one analysis for getting a structure of a macromolecule. So specifically about cryo-EM, typically our main or motivation to develop Scipion, it was single particle analysis. And the problem was that there were many different packages that allowed the image processing of these kind of images, but they seldom talk to each other. So going from one package to another, it is quite difficult, and you have to do many conversions by hand. And we wanted to simplify the comparison of the different outputs. So this is the raw data that you collect at the microscope. So this is how a frame, we call it a frame, how a frame looks like. So they are supposed to collect information, structural information about the protein, but you seldom see anything there because the signal to noise ratio is in the order of 1/1,000. So there is 1,000 times more noise than signal in this kind of images. You take multiple frames of the same region, and then you have to align those frames. And when you average, you increase the signal to noise ratio by a factor 10. And still, the images are pretty noisy, but you may distinguish here some kind of shadows. These shadows are the different proteins that you are interested in. So they are all supposed to be of the same structure, but on different orientations. And then you need algorithms to align the frames, to identify your particles here, and to perform all the steps from the raw data, from the frames to the final structure. And the final structure looks like this. So this is the spike of the SARS-CoV-2 virus. And there are, for instance, mobile parts, and then you want to identify how much these mobile parts are moving in this data. So you can imagine that to go from the raw data to the final structure, it takes quite many steps. So all those steps, and I would say this is universal in data processing, you have to estimate parameters. So I have illustrated it here with the volume that is our final result, but it applies to everything along the pipeline. So you have to estimate a parameter that will be an ideal true parameter that you don't know what it is, but you have to estimate it. And your estimation will have some perturbation with respect to the ground truth. And this perturbation, you can characterize it in two ways. One is what is the expected value of this perturbation. If it is zero, then that means that if this is the ground truth, this green line here, so on average, you will have, if you estimate it once, you will have this value. If you may estimate it another time, you get this one, this one, this one, and on average, you are around the ground truth. So if you average all your estimates, then you should be closer. And if you don't have zero mean, then that means that no matter how many times you average, you will be off by a certain amount. And that happens for the map, that happens for whether something is a particle or not, the alignment parameters of the frames, and all the parameters that you have to estimate along the path. And the other way to characterize this perturbation is by how large are the deviations from the mean. So are you very close to the mean, or you have further excursions of your estimates? And that is typically characterized with the variance, the covariance, and so on. So we have to combine, or we have to estimate millions of parameters. So each one of these images, you have these green circles. You have to estimate whether it is a particle or not. That is a parameter. And if it is a parameter, you have to estimate the pose with respect to the structure. And that is at least 5 parameters. And we have millions of those images. So there will be a fraction of those parameters that you estimate more or less correctly, and that those more or less correct parameters will result into the more or less correct structure, and this fraction I represented here with P. But then the remaining of the parameters that have been incorrectly estimated will result into an incorrect structure that is corrupting your final reconstruction. And I want to illustrate this idea with some examples. So for instance, if you ask an algorithm whether each one of these pixels is the center of a particle, this is what a reasonable algorithm tell. So it is not that this algorithm is crazy, but it is giving more than 1 million possible coordinates. And you see barely by eye that some of them are obviously wrong, while some others you cannot decide whether this is truly a center of a particle or not because of the low signal-to-noise ratio. But for instance, so this is an example of an algorithm that has to estimate a parameter. A point is the center of a particle, yes or no. And then if you use multiple algorithms to do the same task and you see what is in common between them, you see that the number of particles is reduced, or the number of proposals is reduced by half. Many of these errors have disappeared, and disappeared means that the fraction of incorrectly estimated parameters is now smaller, so your final map will be less corrupted. And still there are some incorrect estimations that survived all these multiple pickers, deep consensus that is a neural network that decides whether something is a particle or not, another neural network. So all the algorithms have false positives, false negatives, they have incorrect estimates, and no matter how complicated your algorithm is, still you are never free from this kind of errors. So the fraction, you try to make it as small as possible, but still making it zero is a very hard task. So now you take your particles and you try to combine all those that look similar. So you may decide, okay, this particle, this particle, this particle, they all come from the same kind of particle in the same orientation, and then you can average them, and then you would have these nice classes. But and you do the process for the millions of particles that we have, and some of the classes look very good. You presume that all the images supporting these nice classes should be nice. You see some errors coming from these lines that were caused by the aperture of the microscope. So you presume that most of the particles with these lines go to these kind of classes. You have some errors that you don't know what they are. So probably they are coordinates that have been taken in the middle of nowhere. So there is no particle in there, and you have been able to identify them. And again, this is the answer of one algorithm. So how do you know that this algorithm is correct? And the imperfections of this algorithm will go into the fraction of incorrectly estimated parameters. So now you take what you think are good images coming from good classes, but you use a different algorithm to estimate the class. And then this is what you find. So there are still images that were hidden in the good classes and that still had some problems with the aperture. So what I try to convey here is the idea that executing an algorithm once, it is a very dangerous data analysis approach, because all these algorithms, because of the low signal-to-noise ratio, will make mistakes. And unless you can compare to something else, it is very difficult to identify those mistakes. And that is where Scipion helps a lot, because it allows you to execute multiple algorithms to solve the same problem and to compare their different outputs. So this is another example. This is a 3D analysis of your structure. So this is a ribosome. You collect, I don't know, 500,000 images of this ribosome, and then you try to classify, to identify if there are multiple states of this ribosome. And one algorithm that is not crazy, this one is very much used in the field. So it tells you there are two classes and then some minority class. Probably if you look at the minority class, it doesn't look good. So it is bad particles that have survived all the processing. But then you may try to tell what is the biological story behind these two classes. But because we were worried about the stability of parameter estimation, and deciding whether a particle belongs to a class or not is a parameter. So you run the algorithm 2 other times, and then what you get is totally different. And now, from this story, you may change your mind and say, okay, probably the first execution was wrong for some reason, but the ground truth is this one in which I have three conformations of my macromolecule. The problem is that when you look at which are the fractions of images that have been put all the time together in these three classifications, what you have is a whole continuum. So classifying is particularly error prone, and there could be many errors, and all those errors go into the fraction of incorrectly estimated parameters that is corrupting my structure. The same applies with angular assignment. So angular assignment, for each one of those particles, you have to decide which is the pose with respect to the 3D structure you are reconstructing. And then if you compare the angular assignment of XMIP with the one that is one of the packages that makes this image processing, it is the one that we develop in the lab, with the one of Reliant, they for some of the particles they agree, for half of them they agree, but for the other half they disagree, and they can disagree quite a lot. And they disagree at the level of angles, they also disagree at the level of shifts. And you know that these or you expect that these similar estimates, probably they are coming from this correctly estimated fraction of parameters, but one of the two has to be wrong, or even both can be wrong about this other half of the data. And these wrong parameters will be contributing to the incorrect fraction. And it is not that XMIP and Reliant disagree. Depending on the data set, Reliant disagrees with itself. And this disagreement comes because these algorithms are not deterministic, so they start from a random initialization, so the parameters that they estimate at the end can be quite different between different executions. So the only way to identify which are the parameters that are consistent is by processing multiple times and comparing the different estimates. And that is where Scipion can help a lot. And one of the things that we have developed around Scipion is this validation server. And here is an example of some data set processed by the original authors in their way and processed inside Scipion with all these quality controls. And then you see that the structure coming from the same data is totally different. The overall shape, we could say it is kind of similar, but all the small details are completely different. And the difference between the two is just the processing. So what are your quality controls that you put along your analysis pipeline? And we have constructed a web service where you can upload all your data, and then we ran more than 40 tests. So we we offer you all these quality controls that we have inside Scipion, and we make a report with those. And this report is a PDF, and it is publicly available. It has two modes, so you may want it to make it public, or you may want to keep it private just for yourself and try to correct for the errors. And then depending on how much information you give us, we can run more or less quality controls. So if you just give us the map, we can execute some of them. If you give us a map and two half maps, then we can do more tests. If you provide us with the 2D classes, the particles, the angular assignment, we can do more and more tests. And then in the end, you will get a report where we say we give you all the tests that we have done, and then, for instance, the background for this structure, it seems that there is some problem, and then you may look at the background to see if we are right or not, and take a decision. Oh, I have to change this in my processing, or this report is crazy. I shouldn't pay attention to that. But still now, now you have something to look at. And you can see that if you give us a lot of information, we can give you a lot of feedback. So this is an example of the PDFs. So it can have more than 100 pages where we describe all the problems that your map can have. And this quality control is purely based on Scipion. So Scipion imports your maps, all the data that you have provided us, and then we call the different programs and collect all the results into a PDF report for you. And here is an example of the application of this validation server. So you have 3 different structures of the same protein. They all claim kind of similar resolution, so 3.3, 3.7, 3.8. And here you have the isosurface representation of those proteins. Here you have some slices, and what you see is that even if the resolution claimed is almost the same, the internal details are completely different. So having all these quality controls along your processing pipeline is very important. One of the objectives that we have as a group is supporting cryo-EM facilities. So there are many. So those electron microscopes are very expensive machines, and they are typically centralized into a few institutes in each country. And then some of these countries are run, or some of these facilities are running Scipion for the processing of the data while they are still acquiring. So if there is any problem in the acquisition, you can recognize immediately and then take whatever actions you have to correct it. So your workflows, as I said before, can be very flexible. So you can, if in your facility don't have many computational power, you may just run a very short workflow that only estimates what is the resolution of your micrographs and the stability of your frames, or you may go to very complicated workflows if you have more computational power, where you can go straight away to the final, not to the final structure, but to an estimate of the structure. And along this pipeline, we have put many quality controls, again, to detect possible problems, stability of the gain of the camera of the microscope, stability of the sample, stability of the emitter of electrons, whether you have any beam tilt or a tilt or inside the microscope, if the beam is not very stable, if you have contaminations in your sample. So here are some examples, but we have many of these quality controls that are executed in streaming, so at the same time as you are acquiring the data. And then we collect the output of all these quality controls. We make some summaries that can be accessed by HTML. So if you can publish them in some web server and have the user look at how the acquisition is going on the fly. We also can connect our output to some laboratory information management systems that the different facilities have. So, for instance, this is the ESRF. ESRF in Grenoble, they have their own LIMS that has some summary of how the acquisitions are going. And we also can connect to Empire, that is a public database of cryo-EM data, where you deposit the raw data, or we can connect to some One Data or IROT service where the facility can expose the data to the user so that the user can collect the data from there. And we are now working in closing the loop. So not only analyzing what the microscope is producing, but because we know which are the best quality images, we now can drive the microscope towards those areas that seem to be more promising to have high quality images. And another domain that we are very active is in electron tomography. So electron tomography is a different kind of acquisition mode in which instead of looking at the isolated particles, you look at the particles in their context, in their cellular context. And then again, in the spirit of Scipion, we can combine multiple packages for each one of the steps that you have to go from the image acquisition to the final structure. And you can combine them in any way. So for instance, here you see that we are calling PISEC for some membrane segmentation of your tomograms, but then you are going into XMIP, and then you go into Imantomo, and then so you can combine any package that typically they don't talk to each other, but thanks to the conversions that we do automatically within Scipion, you can go from one package to the other. And here are some nice pictures of the things that you can do in electron tomography. So this is one of the membranes I referred before, and then your proteins are these little arrows here. So and you have to identify them, kind of similar to the particle picking that we did for single particles, and then you can combine all those particles that are now 3D particles. You can combine them into a 3D structure. More visualizers. We are also very active in the development of analysis tools, and visualizers are certainly one of these useful tools to help you understand your data. So in conclusions, there are multiple objectives. If you are at the facility, what you want to process is very quickly, because the images come every 4 or 5 seconds. You have a new image with thousands of these particles, and you want to process quickly but very robustly, so that in a completely unattended way you can have an estimate of how good the acquisition is and what is the kind of structure that you will get at the end. But then at the end, once you get your data home, you want to process them very accurately. And then again, we provide a lot of quality controls and many ways to try to identify which is the fraction of parameters that have been incorrectly estimated. And our main domains are single particles analysis, electron tomography, facilities, processing in streaming. But then I haven't talked today about atomic modeling, but once you have your final structure, then you can try to put an atomic model into the data structure. And once you have the structure, the atomic model, you may try to look for drugs that may interact with your particle, with your macromolecule, and that is the virtual drug screening that Scipion is also covers atomic modeling and virtual drug screening. So the strengths of Scipion as a workflow engine, it is extremely flexible. It allows automatic execution of workflows. It allows interoperability between packages that normally don't talk to each other. You can have all this consensus and quality controls, and it gives you traceability, so you know exactly what you have done, and reproducibility if the algorithms, underlying algorithms, are deterministic. This is obviously not only my work, but the work of the whole group and many other developers, as I showed at the beginning, that are spread around the world. So with this, I'm finished, and I'm open to your questions.